welcome to the Kids First Fall Public Webinar. Thank you for joining today. I would like to remind everyone that it is being recorded. Now that we are in October, we've officially just completed 6 years of the Gabriella Miller Kids First Pediatric Research Program, also known as Kids First. Today, after a brief introduction from us, you will hear about progress on Dr. Sharon Plon's Cancer Susceptibility Project, and Dr. Plon will address questions right after her talk. Then we will get updates from the Data Resource Center before hearing about the recently launched Common Fund Data Ecosystem and how they are working with the Kids First DRC. And we will also share some program updates before addressing common questions and opening the floor to additional questions from you all. My name is Valerie Cotton from the Eunice Kennedy Shriver National Institute of Child Health and Human Development, and I am the Program Manager for Kids First. And I'm here today representing the larger Kids First Working Group, which is overseen by the NIH Common Fund, chaired by leaders from NICHD, NHGRI, NHLBI, and MCI, and includes representation from 12 other institutes and centers. For those of you who may be new to Kids First, the program was initiated in response to the 2014 Gabriella Miller Kids First Research Act, which transferred $126 million from a presidential nominating convention fund to a pediatric research initiative fund and authorized the appropriation of $12.6 million per year for 10 years to the NIH Common Fund, with the first appropriation in 2015. Gabriella Miller, the bill and the program's namesake, was a 10-year-old advocate for pediatric research who lost her battle with diffuse intrinsic pontine glioma, or DIPG, just 6 months before the bill was signed. The vision of the program is to alleviate suffering from childhood cancer and structural birth defects by fostering collaborative research and uncovering the etiology of these diseases, and by supporting data sharing within the pediatric research community, which we believe will accelerate pediatric research and ultimately lead to the development of improved diagnostics and therapeutics for patients and their families impacted by these conditions. Childhood cancers and structural birth defects have profound lifelong effects on children and their families, and several publications, including this one from Sharon Plon and Philip Lupo's group, have revealed associations between childhood cancers and birth defects, suggesting that there are shared genetic pathways underlying these conditions. But since individual childhood cancers and structural birth defects tend to be relatively rare, there was a need for coordinated efforts to build, combine, and study sufficiently powered data sets. To respond to this need, starting in 2015 and continuing through this year, we are whole genome sequencing childhood cancer and/or structural birth defect cohorts. And since 2017, we have been developing the Kids First data resource to facilitate and empower analyses of these data sets. And during the program update section at the end, I will tell you a bit about our plans for the remaining years of the program. To date, 40 childhood cancer and structural birth defects cohorts have been sequenced or are somewhere in the sequencing pipeline, and 14 of these data sets are accessible to the research community. The data are or will be shared through the Kids First Data Resource, which is a cloud-based platform designed to empower and accelerate collaborative pediatric research. There are multiple components of the resource, but the two most important to this community are the Data Resource Portal, which is where researchers and the public can view and visualize Kids First data and discover which data sets you want to learn more about or analyze based on your scientific questions. And the cloud-based workspaces in Kvatica, which is where researchers can pull together and co-analyze multiple disparate data sets. Of course, you'll hear more about this from the DRC's presentation today. More than 1,700 users from across the country and the world are accessing the portal, and more than 150 data access requests have been approved for external researchers to analyze whole genome sequence data from the 14 available data sets. And researchers are analyzing these data with support from a variety of NIH grants. Although Kids First does not fund analysis grants out of its appropriated budget, institutes and centers from across the NIH support this investigator-initiated work through a variety of mechanisms, including an R03 that is specifically designed for supporting analysis of Kids First data and associated pipeline or tool development. And we are happy to see that many of you have been successful in competing for these awards. And discoveries are emerging from these analyses, such as uncovering new genetic causes of childhood neuroblastoma, identifying genes or regulatory networks that are important to the development of the diaphragm or the heart, and identifying genetic variants that lead to craniofacial anomalies in different ethnic populations. You've heard from some of these investigators in the past, and we will continue to highlight these findings in future webinars as well. And without further ado, allow me to introduce our featured Exo-1 speakers for today. Dr. Sharon Plon's research has focused on understanding inherited susceptibility to childhood cancer, as well as other congenital anomalies. She's a leader in high-impact initiatives such as CSER, or the Clinical Sequencing Exploratory Research Project at Baylor, and ClinGen, and serves on NHGRI's Advisory Council. In the spring, you heard a bit about her from her student, Owen Hirschi, and how he's used Kvatica to develop and run workflows to analyze this Kids First data set, and I believe you'll hear a little bit about the progress of his structural variant analysis today. So with that, I'll help unmute you, Dr. Plon, and I will hand over the presentation.
Great. So you'll let me share. Well, thank you. That was actually a great intro. I didn't know you were going to do that, although I didn't realize how many years we were in already. So I did just want to comment, since you gave the history, that I had the honor of meeting Gabriella Miller's mother, who early meetings when Kids First was getting underway, and really just talked about the importance of this work. So great to hear the overall progress. So that should look correct. Does that look right? Yes, Valerie?
Great.
Okay, so I'm going to talk about identifying novel cancer susceptibility mutations from unselected, and I want to emphasize, that'll be sort of a theme I emphasize, childhood cancer patient and parent trios from the Basic 3 study. So I'm going to give a little bit of background about what did we know about these families based on our CSER project. That's where they came from. And then what did we hope to learn with whole genome sequencing? And then Owen's going to give an example of a very recent, totally unpublished finding that we're excited about pursuing. So just a disclosure that I'm on the Baylor Genetics Scientific Advisory Board for their laboratory. So really, there have been multiple different approaches to understanding the genetic basis of childhood cancer. And I'm going to briefly talk about what's been going on most recently with regard to our Basic 3 study, and I won't talk about the subsequent study. I think you're all aware, and is emblematic of Kids First. Most of the Kids First projects are sequencing large cohorts of specific pediatric cancer patients. So the neuroblastoma discovery that just got mentioned came from one of the Kids First neuroblastoma cohorts. And we won't talk about today, but another area where we're really learning more about genetic childhood cancer is from precision oncology trials that are really designed for treatment trials, but are now beginning to do matched paired germline and tumor sequencing. So the NCI Children's Oncology Group Pediatric Match Trial does do a germline panel along with the tumor panel. And I think they are looking to now, on a research level, do whole genome sequencing from that cohort as well. So as was already mentioned, this project started as part of an NIH initiative called CSER, the Clinical Sequencing Exploratory Research Consortium. It's now still CSER, but now we're the Clinical Sequence Evidence Generating Research Consortium. So I just want to say that the original project was a very large collaborative effort across Baylor, sequencing, ethicists, geneticist, and Will Parsons, who is a pediatric oncologist, many of you know, who's been my partner throughout these studies. So I just want to spend a little bit of time on this study because it really just highlights who are the patients we're talking about. The Basic 3 trial, which started enrolling in 2012, enrolled sequentially newly diagnosed unselected patients. So they had to have a brain tumor or a non-CNS solid tumor. The family had to speak English or Spanish. Those were the only, and it had to be a malignancy. So if a patient had a lipoma or a benign neurofibroma, that was not included in the protocol. But otherwise, we took all comers, and I'll show you what that looked like in a couple of slides. We isolated frozen tumor and blood from both the patient and both parents. And we did CLIA-certified whole exome sequencing in a clinical laboratory that was a joint effort of the Human Genome Sequencing Center and the Baylor Clinical Diagnostic Lab at the time. And we generated a germline exome and a tumor exome report for each patient. If a mutation was found in the child, then it was validated in the parents, but we did no sequencing of the parents otherwise. The results went back to the electronic medical record. They were disclosed to the family by their oncologist and a study genetic counselor, and we followed them. And the goal was really, and A, could you do it? Remember, this was starting back in 2012. And could do it, what were the overall results of incorporating germline and tumor sequencing into pediatric care? So there are a couple of different, and just to say the methods were all published in our original publication. We're halfway through the study, but I'm going to give you the final results, which sadly have not yet been published. So there are a couple of ways to think about diversity, and I think this is something we don't talk that much about at Kids First and probably worth looking at, which is what is the underlying diversity of the patients that we're enrolling in these cohorts. And so we were very proud of the fact that, as many of you know, Houston's a very diverse city. And this study, which was published, and then these numbers are actually updated once we completed the study, or close to completion of the study, was that there was really no difference in the decline rate. So the patients who entered the study represented our population. And you can see here the study overall, and the final study, I believe, was 48% Hispanic. And it was about 10% African American. And then there was the majority of the other patients were non-Hispanic whites. But it was a diverse patient population underlying their ancestry, underlying some of these findings. Another way to think about diversity, and this is particularly relevant to the study, that to people who want to use our Kids First data, is this is very much unlike most of their Kids First projects. We took unselected patients, so Will took the first about 150 patients and made these beautiful pie charts. I don't think he ever completed them for the whole 282 patients. But you can see we have a wide variety of malignancies. We have both the ones you would expect that are more common, neuroblastoma, Wilms tumor, but we had individually very rare diagnoses, and I'll comment on some discoveries we made out of that. And that's true of both the CNS and non-CNS patients. And then finally, although I won't talk a lot about this, was the diversity of what we returned. So every patient had a tumor report with every somatic mutation that was in a coding region. And then they had these 5 classes of germline results. The one that will be relevant to our discussion today are the cancer findings. So I want to just briefly go over the cancer findings and then what our plans were for Kids First. So of the known cancer genes, so these are molecular diagnoses. So these had to be known genes. What is it? About 10% of the patients carried a germline finding. And our surprise was, first of all, even though it's only 27 patients, it represented 19 different genes. So although P53 was more common, it was not the major outlier. We saw it 3 times, as did we see VHL. But we saw a wide variety of individual genes, many of which were known to be associated with the tumor, DICER1 and a pulmonary pleuroblastoma patient. But surprisingly, almost 40% of these findings were in genes that were not to be associated with the patient's tumor. And this has been quite interesting. We're not alone in this early. St. Jude published their large series a few months before ours, and they also reported BRCA1 and BRCA2. As I think many of you are aware, this has now been looked at in several additional cohorts where there were large enough cohorts to do sort of case control analyses, including our study that's not yet published, but soon to be on rhabdomyosarcoma. And it does seem that BRCA2 particularly may be overrepresented in pediatric cancers, and I think that's something interesting. Another thing to think about when you're analyzing Kids First data, which was very sad to me because I spent much of my early career studying rare recessive disorders like Rothman-Thompson syndrome, is that almost all studies that have been published so far, biallelic mutations in recessive disorders are extremely rare. So one of our 300 patients had a disorder that I'm going to talk about in a minute related to hepatocellular carcinoma. The St. Jude study, again, I think of their 1,100 patients, they had 2 or 3 at most. And I think it's important when you're looking at the data to contrast this with the fact that single pathogenic variants in recessive cancer genes are quite common. So we found about 0.5%. I think St. Jude found around similar numbers, maybe 7%. And the significance of those single pathogenic variants is really unclear. Again, there was a study recently suggesting that single RECQ-4 variants in osteosarcoma may be significant. But again, when you're analyzing the Kids First data, you will find a large number of single pathogenic variants in recessive disorders. But the contribution of truly biallelic, a recessive diagnosis in pediatric cancer seems to be very, very low. Okay, so why do whole genome? And why do whole genome from such a heterogeneous data set? And I will say the original reviewers of my EXO-1 application had a similar number of questions. So we were delighted when we were selected despite that concern. And I just want to give 2 examples that came out of our exome data of why it's important to remember that even if we are sequencing heterogeneous data sets, and we may only have a few patients with that type of tumor, that if you do find something, there are other data sets or other rare patients that you may go to confirm it. So these are actually 2 papers that came out of Basic 3, both from single patients in the study. So I mentioned already that we had a single patient with a biallelic. This patient was homozygous for a frame shift in TJP, tight junction protein 2, and had hepatocellular carcinoma. And through one of our collaborators at Texas Children's, Dr. Lopez Terrada, who studies pediatric brain tumors, and Dr. Feingold, they identified that they had a collaborator who actually also had a patient with hepatocellular carcinoma and had this otherwise extremely rare inherited disorder of liver function. And in fact, we were able to combine the data, look at the pattern of pathology in the livers and in the tumor, and publish this. And subsequently, actually, several other patients with TJP2 deficient cellular carcinoma have been reported. On the right is probably an even more remarkable study. We had a single patient with clear cell sarcoma of the kidney, actually had a stop loss predicted by the exome. But Angshan Roy, who was reading them out, was sort of surprised by that. He actually pulled the reads, and actually it became clear that in fact it was only a stop loss because there was an internal tandem duplication at the last exon, at the last codon. And he actually was able to pull tumors from the TCH pathology collection and actually showed, I think it was like 8 of 10 had internal tandem duplications of B-core, and then was actually able to confirm that in the target data set. So again, an example of a single unusual result in one Basic 3 patient that then led to follow-up studies. So I think really that, in a way, was our justification for saying it's worth taking this data further. All right. So to kind of quickly fill the goals of our, the goals of our Kids First project were to utilize the rich existing data set from Basic 3 to make further discoveries using whole genome sequencing of the proband and the parent trios. So just so people are aware, because this is available to you, the germline exomes of all the probands have been submitted to dbGaP. The tumor exomes from the majority of the probands, I think 70 or 80% of the probands had tumor exomes, have been submitted to dbGaP or in dbGaP and being used by others. We actually do have RNA-seq for just a subset of the tumors. We did, though, have DNA from the parents and the patients. And these blood samples were very high quality DNA. And that actually gave us great whole genome short read data. And as you'll hear, was the reason we applied to be part of the long read. And we actually also have lymphoblastoid cell lines. So every patient and every parent in Basic 3, we endeavored to make LCLs. So we actually can grow cells, we can do functional assays, we can do splicing studies as needed. So how are we approaching the Kids First analysis? Obviously, we're doing trio sequencing. And we prioritized complete trios with adequate DNA, obviously. We did have some patients with more unusual histories, multiple malignancies, a few patients with birth defects. The study that I did with Philip Lupo, I think it'd be great to have Philip come and talk about that study. That was a population-based study and not based on basic 3. We did actually include a few solved cases. So, for example, a parent with a P53 mutation, because we actually wanted to look at how many did the child have. And we did that analysis. I have to say, I don't think there was anything remarkable that came out of it, but it was one of our original goals. So generally, our analysis has focused on both single nucleotide variants and structural variants. We've prioritized de novo variants in both situations. However, we have also analyzed known cancer susceptibility genes, looking for any missed variants that might have been missed from the exome. And more recently, we performed Splice AI, which is a relatively new artificial intelligence algorithm to try to predict potential missplicing events. So we've also done that with all of our Kids First data, and so far that hasn't yielded anything too interesting. So just briefly, so I can turn it over to Owen, talk about structural variants. We did de novo SNP calling. I will say this is where the Kids First team has been extremely helpful. We've already this platform on our Human Genome Sequencing Center data, and we've been using Platypus, which was a paper that was published in Nature Genetics a couple years ago, which was specifically designed for trio analysis. And so we were able to work with the Kids First team to bring Platypus as a tool into Kvatica. And so obviously our prioritization was pretty standard. We prioritized, in this case, de novo coding variants, although I will say we have gone back and looked at all known cancer susceptibility genes, see if we missed anything. We determined, obviously, the predicted functional effect of the variant. We ranked them based on things like CAD, and we also did sort of hand mining of the medical literature for them. And we got kind of the typical, got genome-wide, 60 under it for most of the trios. There's one trio that has more de novos, which we've tried to explore and we don't have a good explanation with, and not surprisingly, 0 to 4 de novo coding variants. So this data has been interesting. I would say the main de novo variant from all cohort that we've been looking at is heterozygous missense variant KMT5C. It's rare, it's in a highly conserved functional domain, has an extremely high CAT score and a high REVEL score. The patient has ependymoma, which, as you'll hear, Owen is interested in generally, and has an interesting structural finding, and is an example of a variant in a chromatin remodeler. And chromatin remodelers play a huge role in pediatric brain tumors, so that's particularly interesting to us. So that's sort of where the SNV data stands. And then Owen, I think, is the next slide, is just going to talk briefly about his structural variant work. He'll repeat a little bit of what he said in the spring for those who missed his talk, but then he'll show you kind of an interesting new result. I can just advance the slides if you can unmute him.
Yeah, that would be great.
Okay, we can hear you.
Okay, great. Well, hello all. I'm Owen Hirschi, one of Dr. Plon's graduate students at Baylor College of Medicine. And today I'll be talking about the structural variant analysis in the Kids First Basic 3. So a little background on structural variants. They're commonly reported in cancers, and some are known to play critical roles in said cancers, such as the Philadelphia chromosome translocation in leukemia. In the Plon lab, we've actually recently reported on germline structural variants in pediatric cancers, such as a balanced inversion of the APC gene in a pediatric hepatocellular carcinoma. And this was actually discovered through RNA sequence analysis. However, identifying these germline events in short read whole genome sequencing is challenging. Since 2010, over 40 programs have been created to identify these structural variants from short read whole genome sequencing. However, they were primarily built for identification in tumors, which requires the elimination of germline events in the results. And each of these individual programs have low sensitivity and a fairly high false positive rate. So that's where we believe we can kind of create a better pipeline to identify these germline events. Next slide. So for our pipeline, we actually want to use 5 different structural variant callers. So these are all available on the Kids First Kvatica platform, and they're mentioned below. And so what we do is we run these 5 different structural variant callers on each member of the trio and generate a list of variants identified per individual. Then we use various sliding filters and visualization methods to dwindle down which are false positive, which are true positive. And so what we found is using multiple structural variant callers has allowed us to increase sensitivity and specificity in variant identification. And this was tested on an independent childhood cancer cohort. Currently, this pipeline is built for the identification of de novo structural events, but we're now beginning to tune it for inherited variants. And so I want to talk a little bit about how we use this pipeline on Kvatica. So Kvatica has been very useful in that you have 2 real modes of operation to run tools. First is through application base, and next is through terminal base. So for the application, we're allowed to run up to 80 parallel tasks at once, which in comparison to your local system, you may be able to run up to 8. So if you think about it, when we run 5 different structural variant callers on 165 different samples, that saves on quite a bit of physical hours when completing this. And these applications are tunable just in the same way you would tune them locally. And then once we generate those variants, we can analyze them in the interactive analysis cruncher feature of Kvatica. So this allows us to run complex bash scripts for filters and visualization tools and allow us to identify which are true positives. And it's very similar to what a user's terminal would be running these bash scripts. Next slide. Yeah, so just to talk a little bit about the filters we do use, we first filter out those structural variants that are found in poorly mapped regions of the genome or low complexity regions. And right now, we've just looked at de novo events. So those are only occurring in the child and not in the parents. Then we filter against a healthy population to determine for rarity. And then finally, just to determine these are true positive, we visualize the breakpoints in IGV or integrated genome viewer. And the outcome of this manual visualization process on our 54 program trios, we identify anywhere between 0 to 1 de novo structural variant per trio. This fits in line with published literature on what you would expect to find in general population. So we identified 10 deletions, 7 duplications, and 0 inversions in the 3 cohort. We're currently looking to identify these translocations, but that's still a work in progress. So a couple variants stick out, and so the first one I'm going to talk about is this germline de novo heterozygous 20 kb deletion of NF2, which is a known cancer predisposition gene, in a meningioma patient. So what we're looking at here is the visualization of next generation sequencing data in IGV, which shows where reads map to on the genome. So we identified this deletion, which can be denoted by the drop-in coverage you see in the proband. This deletion occurs in exons 5 through 9 in the NF2 gene. And what's kind of interesting about this mutation is, unbeknownst to me, it was previously identified when this patient was first diagnosed. This was done through clinical sequencing of NF2, and they identified it as a potentially somatic mosaic event. And so we can kind of show that this is also potentially mosaic, as we only see it in 6 of the 40 reads in IGV. And this is important to us because it shows that our pipeline can find true positives as well as potentially mosaic events. Sorry, next slide. So the next one I want to talk about is a particularly interesting variant to me because it's a germline de novo heterozygous 8 kb duplication of the prostaglandin reductase 2 gene in an ependymoma patient. So what we see here is an increase in coverage in this highlighted region within the proband. And from the whole genome sequencing data, identify this as a head-to-tail tandem duplication. And one of the more interesting facts about this is it overlaps the promoter and first exon of all transcripts of the PTGR2 gene. So PTGR2 is actually a putative oncogene in brain tumors, and it's expressed. And the reason this is interesting as well is because this increase or this duplication may lead to an increase in transcription. Slide. So on initial analysis of this variant through ENCODE, we can see this hypothesis may be correct. So what we're looking at in ENCODE is whether, or they identify regions where there's open chromatin, so it's able to be transcribed, whether there's active chromatin marks nearby, and whether there are regions in your location that may affect the 3D architecture of the genome. And using that, ENCODE is able to predict whether certain regions can act as promoters or enhancers. And so what we see in this duplicated region is that there's duplication of 5 like regions, which would lead us to believe that there will be a substantial increase in transcription. And as Dr. Plon mentioned earlier, some of these basic 3 samples have undergone additional research studies, and this sample is one of those. So we are just beginning to look at RNA-seq data to further investigate whether this variant does play a role in the transcription and potentially implicating this child's cancer. From all of this, we've shown that we've developed a pipeline with the aim to better identify these germline de novo events. And we have identified a few particular de novo heterozygous duplications that we're currently following up on. And we've greatly benefited from the additional research studies involving both tumor and germline sequencing of phase 3, which doesn't stop with the whole genome sequencing. Yeah, so as Dr. Plon mentioned earlier, we've been selected for long read sequencing of basic 3. And this is greatly helpful for the identification of structural variants, because in short read whole genome sequencing, there's a problem with poor mapping. They don't map very well to low complexity regions of the genome. And so by performing long read sequencing, we'll be able to identify more structural variants. And so we plan to use a similar methodology to what we use in our short read pipe, but instead we'll only be using one structural variant caller. That's called Sniffles. It was developed here at Baylor College of Medicine in the Human Genome Sequencing Center. We'll then prioritize de novo variants and those that do not occur in a healthy population. So the goals of all of this study is really to compare the 2 results of this structural variant analysis. And it will enable us to identify the limitations and accuracy of our short read structural variant pipeline, as well as structural variant calling in general in trio studies, and as well identify causative structural variants only found via long read sequencing. I want to thank Gabriella Miller Kids First again for this opportunity and this opportunity to talk.
Great. I think that's just questions. Yeah.
Great, thank you so much. Dr. Plon, you already have a question from Dr. Spector. Do you see it, or do you want me to read it out?
Yeah, I don't see it, so that'd be great if you read them out.
Sharon, are you surprised that biallelic recessive variants are rare in pediatric patients? ? My impression is that they are usually accompanied by clear phenotypes, so their prevalence had already been established prior to the sequencing era.
Yeah, I mean, Logan's right about that. I think what was not known is that there are hundreds of them, right? So each of them are individually rare. So like Roth, right? Lisa Wong started with me, and now is her career, and I think at Texas Children's, we've still, in those 20 years, maybe 2 or 3 additional cases. So we know they're rare, but there are dozens of DNA repair disorders. And the question was, if you sort of add them up, would there be more? I think they don't all have phenotypes. So ataxia-telangiectasia, for example, eventually have a phenotype, but children can definitely develop cancer before they ever develop ataxia or telangiectasia. So, and that's definitely true of some of the other DNA repair disorders. So it is true that we knew that each one individually was rare. I was surprised that, for example, that there weren't more undiagnosed Fanconi anemia patients, because that's another one where the prevalence of the birth defects is highly variable. And there are a lot of patients with Fanconi anemia that might not have been detected to have that disorder before they developed cancer. But yes, I mean, the gestalt is that they are rare, but I thought they might have made up at least a few percent. And more of these studies are suggesting it's probably less than 1%. On the other hand, I should say in our study of rhabdomyosarcoma, we were surprised to find that about 1 in 5 of the 500 patients appear to have Costello syndrome. So that's an example where what we thought was really, really rare may in and of itself make up 1% of rhabdomyosarcoma patients. And so it was a little bit more that kind of data I was expecting than what we did find.
Thank you. You have another question from Laura Eagles. What data sets did you use for filtering against control populations for the SV study?
Owen, I'll let you take that if you haven't disappeared.
Yeah, I'm right here. So for the structural variant, we are initially just doing the other unaffected parents for our initial healthy control population. But I'm sure as many of you know, NOMAD recently came out with a large structural variant pipeline on what appears to be non-cancer affected patients. So we're also comparing those variants one-on-one to that, but the initial filtering is only against other unaffected patients. One of actually the nice things about the long-read pilot is Genome Sequencing Center here at Baylor does actually have cohorts that undergo the same long-read sequencing that our samples will also undergo. So will actually serve as a very nice healthy control in that sense as well.
And for your finding, now that you've been able to look in NOMAD, has that been called?
No, it's not. So it's completely novel.
Okay.
You had another question that, oh, go ahead.
Oh, yeah. So how many patients are you aiming? So we are aiming for roughly the same amount who underwent short read whole genome sequencing. We are limited in part by the amount of DNA that's required for long read sequencing. It's quite a bit more. But from initial estimates, we can get roughly, I would say, hoping, fingers crossed, 60 of the 67 we originally sent for short read whole genome sequencing.
Well, and I should say, because Valerie will probably correct us, we've been approved for the pilot, which is much smaller, and then we are likely to be approved for the longer one, assuming the pilot goes well.
Yeah, that's right. And I will talk a little bit about this overall later in the webinar, but all the numbers are sort of TBD, you know, because of the QC at play. But yeah, you guys are targeting a pretty large sample, so we're excited about it.